Thanks for joining me on my ride. That was a paper from Jozsef Csicsvari's lab in Austria. This lab is known for all kinds of crazy experiments, and the first author, Igor, has done something really special here. I think for a very long time, since the David Reddish lab and other labs have found preplay and replay in the hippocampus, people have been hypothesizing about how we can mess with these ensembles and see what role they play in memory. We've tried a lot of different ways to interrupt the shortwave ripple, and in doing so, we haven't been specific enough, I think. And this paper really uses brain-mind interface in a spectacularly clever way to try and find ensembles for two different environments and selectively inhibit one ensemble versus the other. And so, Ruby, and sit. So, sit. So the idea is then that what they do is they train the animal to find the food in the maze. Then they let the animal sleep, and during sleep they use the brain-mind interface to detect specifically replays of the target environment. And when they find those replays, they mess with them by stimulating the whole hippocampus and disrupting it. It's not a perfect disruption. It's not something like the Tonigawa lab might do with disrupting specific cells. But on the other hand, because they're using brain-mind interface to find out exactly what preplay and what replay is being done, then I think in this way they're more specific because they're doing it in real time. It's not to say that the Tonigawa's lab's techniques aren't cool, but they would still require the brain-mind interface in order to specifically nail the shortwave ripple when the right replay is going through. And I think this is best characterized by their last figure, where they show that place cells which have fields in both environments, the field that's in the target environment is the only one affected by the experimental treatment during sleep. And that's pretty spectacular. So all I can say is congratulations, Igor, and congratulations to the Csicsvari Lab for what I think is a paper long time coming, and I'm really glad you guys nailed it. If you liked it, like and subscribe. I've got at least 2 more big hippocampal papers coming this week or next. There was a really big paper from Mike Hasselmo's lab that just came out in Science Today that completely blew my mind. It's actually in retrospinal cortex and shows some very interesting things. I need to take that one on. And there's also a Matthew Larkram paper that recently came out showing the decoupling of apical dendrites from the soma in relation to anesthesia, which is also a very exciting result. Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC. This is Journal Club on a Bike. I have a great paper tonight from the Csicsvari Lab in Austria. They do all kinds of crazy stuff there. They are working in frontal cortex and in hippocampus. And wherever Jozsef Csicsvari goes, questions fall, and today he does something that follows up nicely on the Lauren Frank Lab's recent offering and follows up very nicely on the Reddish Lab suite of papers regarding preplay and replay, and sets us up for something really exciting. It's something that I think a lot of people thought was possible for quite a while when they saw replay, but a very tricky experiment to pull off. So essentially what we're looking at here is that lots of different papers have played around with sharp wave ripples during sleep. And when you analyze the sharp wave ripples that occur in the hippocampus during sleep, what you see is that ensembles of place cells in the order in which they were activated by previous experiences get replayed. And that replay is governed through the sharp wave ripple. These ensembles replay through sleep. And a long-standing question was, well, if we mess with these sharp wave ripples in the hippocampus, what happens to memory? And I think a vast majority of folks have thought that this has to do with reconciliation of memory, long-term storage. Back in the old days, we used to talk about the transfer of the memory from the hippocampus to the cortex. We don't really talk like that anymore, but this was the kind of theories that were going around with what does the sharp wave ripple do? What are these network dynamics and why are they there? And so figuring out how to perturb these network dynamics to see how they affect memory has been done a few times with electricity and even with optogenetics. But the tricky part is to differentiate what's going on in an individual shortwave ripple. So if you just inhibit all of the sharp wave ripples in the hippocampus, you're just going to inhibit all of the come on, folks. Seriously? No way in hell. He would be blocking all of the memory processes attached to the sharp wave ripple, right? And so what this paper is going to do is try to set up a brain-mind interface where the computer can look at the activity recorded in the CA1 and decide whether a highly synchronous event, which usually leads to a shortwave ripple in this big paper, if one of these events is for one environment or for another environment. See, I don't understand the way that this bus is driving. Did he just let people out in the middle of the street and make that guy stop? I mean, that's just crap driving there. And so how's this going to work? They're going to do something very similar to what the Reddish Lab did. They have a circular cheese board, lots of holes in it, and then every day the rat comes into the cheese board maze and has to find out where the food is. And once they're good at this task, it's only the very first trial where you can see that they don't know where the hole is, and then after that they pretty much go directly to the hole. And the authors state that about anywhere from the first and third trial, they're pretty much straight on to the hole in both the target and the control environment. So there's 2 cheese board mazes that they learn every day, and those cheese board mazes are the cheese board maze environments are learned new every day. They're the same environment, but the location of the food in each one of those in each environment is changed every day. And so their idea is that the animal will store the location of the food after 5 trials, and then they're going to put him in a flower pot and let him sleep. But they leave him hooked up to the 128 channel recording device, which can also stimulate with 4 optic fibers. I don't know if the device is the same or not, but they have 4 optic fibers, and they're expressing chanorodopsin, right? Yes, chanorodopsin. No. Yes. They're expressing in CA1. So what they're going to try and do is silence CA1, but of course they don't get arch in every cell, and so there is a diversity of effects that it has on the population of pyramidal cells as a whole. So in the first figure they're showing you a diagram of the basic plan, which is a learning day, and that's 5 trials on the same day, and then some time in the flower pot. I don't know if it's hours or not, but a rest. And then they do a probe trial. Holy shit! They do a probe trial, and what the probe trial is a trial in both environments where the food well is not baited, but the animal reports its perceived memory, or the quality of its memory, by how many times it crosses the right hole, how long it dwells at the right hole, and the overall path length that it expends trying to find the food, with the idea that the longer that path length is, the less confident the animal is about where the food was last. And then they put him through a new learning paradigm where again the holes are filled, and we see that in the first 2 trials you can tell whether or not they really remember or not, because the control environment that is not messed with, when those ensembles are not disturbed during sleep, then the knowledge of the control environment will be evident in the behavior of the animal, whereas they will go right to the hole. But in the first 2 trials, after the probe trial, and after they've disrupted the ensembles in the target area during sleep, then you can see that during those first 2 trials, again you have the same thing. You have the same issue where in the control environment the animal is just going to demonstrate that it hasn't forgotten where the hole is. And in the target environment you will see that the animal scrambles a little bit before remembering or restoring, if you want, its knowledge of where the food is baited. And so they're going to look at a few key time points. They're not going to look at every trial. They're going to look at the probe trial, then they're going to look at the first and the last of the second group, and to show you the learning changes. And so the key is to remember that whenever the animal is in the pot, they have this brain-mind interface that's going to try and detect highly synchronous events before the shortwave ripple, and then turn on the arch and mess with the sharp wave ripple that is going to replay the target environment and not the control environment. And so that's already happening in figure 1. But now in figure 2, what they're going to show you is what they see from the perspective of unit recordings at 2 different time courses. One is at theta, guy in a Tesla reading his phone. One is at a time course of theta, so that's a pretty, high, a lot of time shown there. And then the one on the right is at the sharp wave ripple frequency, and so there you see the time is much expanded, and you see that the highly synchronous event is detected a few milliseconds before the actual shortwave ripple shows up. And this is another argument that they're doing this really exceptionally well, that within about 50 or 100 milliseconds of the start of a replay sharp wave ripple event, they are already able to identify the highly synchronous event which precedes it, and then the brain-mind interface can decide whether or not to turn on the green laser and inhibit. Don't run, it's Friday. Whether they can turn on the green laser or not and inhibit that ensemble. And so what they're showing you in figure 2 then is the kinds of effects that they see. In figure 1 they also showed you the broad kinds of effects. Remember, if they turn on arch, the pyramidal cells that are expressing arch will be inhibited. But there will be other pyramidal cells that might be disinhibited by the inhibition of surrounding pyramidal cells. And so they see that, and they also see inhibition and disinhibition of inhibitory neurons. I don't think they show both sides of that though. And that's figure 1. Then in figure 2, they go immediately to showing you at 2 different time scales what they see before and after the detection of the highly synchronous event. And that's the trick, because these next couple figures will always be divided in half, and for the control environment, dividing in half doesn't really mean anything. It's just showing you when the event is detected. The event is always detected at the middle of this graph. And so they show you the 100 milliseconds before and after the detection of the event. And what they want to show you is that when the light comes on, some cells are inhibited, some cells are disinhibited, but the effect of the light, that would be on the right, in the target environment, is the disruption of whatever replay was going to happen. That's figure 2. So what figure 3 then looks at is the effects of this on their performance in the cheese board maze. And it's going to specifically show you some paths, so you can look at the raw path of the animal to see that they learned. And then the first trials after the sleeping period is when the target environment's memory is disrupted. And it was disrupted during sleep, but the readout they get is from the probe trial and from the first and last second learning trials. And so there, what they show you is the effects of firing rate and the changes in firing rate before and after the detection of the event. So here again, what they're showing you is that the firing rate of the control cells in both environments doesn't really change, although you would expect it does change in the target environment, because like I said, if there are place cells that code for both environments, then when the place cells that code for both environments are firing for the target environment, we're going to turn them off. Just a munch. It's okay. I don't mind munches. And they also show in the far last figure is a total path length, and you see that after the memory disruption, the total path length in the target environment is quite a bit longer, because they're exploring, they don't know where it is. And so what figure 4 is going to look at then is individual cells and try to give you an idea of what they expect, and then give you an idea of what they saw. So again, figure 4 is trying to show you what they expect on the bottom. So here they show you the detection of the event in the control environment on the left, and detection of the event on the control environment on the target environment on the right. And then they show you a little cartoon underneath that shows what they expect. Oh God, please just drive. What they expect the cells to do. It's not real data underneath there according to the caption, but it's just what they expect the cells to do. And then the actual firing rate that they measured is in the bar graphs, I believe, if I'm getting this figure correct. And I think I am. So what you should be seeing here is sort of what did you expect to happen? And they're also showing you, that's all I also have to say, is that they're analyzing the cells that were inhibited and the cells that were disinhibited separately. So they do that also in figure 3, I believe. So the trick here, figure 3 shows you that the erasing of the memory has an effect. Figure 4 shows you that there are definitely effects on individual cells that they expected, and those effects are measurable, quantifiable across experiments. So after they show you the rates in figure 4 and how the rates change before and after the detection of the event in control versus the target environment where they try to interfere, is now they're going to look at ensembles. So cells that fire together in one environment or another, and to try and demonstrate that before and after the detection of the event in the control, before and after the detection of the event, they show you what's going on and what those various ensembles do together. And then in the target environment, they show you how hypothetically how the firing of ensembles should be altered. Again, the raster plot underneath the initial figures is just a cartoon. The data is the bar graphs. Again, just try to take a look at how these pairs are firing. There might not be bar graphs in this one. I can't remember now. Sorry. But this one looks at how the ensembles fire together and shows that co-firing is affected by their light. And so they show a specific kind of disruption, right? Which is a nice additional piece of data. And then figure 6 is pretty tough to read unless you're a place cell guy. I mean, if I had any critique, I find these figures they're tough to penetrate. They're organized well, but somehow I find them not intuitive. But the 6th one is going to look at place fields of the cells that they record from during the behavior times, during the learning, initial learning, the probe trial, and the relearning. And they look at how place cells are disrupted in the target environment and return during relearning, and how cells in the control environment are not affected. Interestingly enough, they show cells that are active in both environments, and the place field in the target environment is affected, and the place field in the control environment is not. So that's a pretty home run result there. What a great paper. Congratulations, Igor. I think I've been waiting for somebody to do this one for a while. Awesome.